In this series of videos, you will learn how to complete an RT-PCR reaction using an Applied Biosystem Step 1 Plus thermocycler. Before beginning, there are some preparations that need to be made. The primers used in PCR must be optimized and the best concentrations found before the experiments can begin. Primers are specific series of nucleotides that bind to DNA during the replication process. When the double-stranded DNA splits at the cleavage fork, primers attach to the two now single-stranded DNA segments. DNA polymerases come in and copy the segments that the primers are binded to. Because of this, properly designed primers are of the utmost importance, as is having both forward and reverse primers present. You also need to choose a fluorescent marker. There are two main chemistries used to detect the PCR process using real-time PCR instruments. One is TaqMan probe, and the other is cyber green dye, which detects all amplified double-stranded DNA. The choice of marker chemistry does not change the basic procedure. All plates that are run must contain an internal standard or housekeeping gene. This gene will be the benchmark for amplification of the other genes. We will use 18S RNA, which is a common housekeeping gene because it is a mitochondrial RNA. This means it is rather abundant, as well as stable. In this graph of PCR results, the red line is the 18S RNA. You can see that it crosses much sooner than the other genes being tested. It should always cross the threshold at the same point in the cycling process. The last important consideration is that each gene needs both a PCR control blank and an RT control blank. The PCR blank has only the master mix and molecular biograde water, and the RT blank is the control blank from the cDNA step and its respective master mix. If there are errors, this can help you determine at which step in the process the problem occurred. The other test that can help you determine where an error occurred is in the melt curve portion of the PCR run. All of the melt curves for a particular gene should match up. If they do not, that signals contamination or an error with the primers. The last important note is that pipetting accuracy is of the utmost importance, especially in the preparation of the PCR plate. To learn how to make the master mixes, please watch the next video.